Hi class, welcome to Unit 4. The topic is isolation and quantitation of bacteria. Before we get started with Unit 4, I do want to advise you to review the study guide. So it was posted in an announcement yesterday. And also to review the updated Unit 3 slide. So as I was writing the study guide, I realized that the Unit 3 PowerPoint didn't have all the detailed information required. So please download the updated Unit 3 slides, and I'm going to go over those additional slides in this recording. So in Unit 3, we talked about biochemical reactions, and it started with carbohydrate metabolase reactions, and it talked about fermentation test, and there was a slide dedicated to phenol red broth and how you are going to interpret the result. There was a few additional tests mentioned, and then it was only a line talking about the MRVP test. So I want to explain a little bit more about this. So MRVP stands for methyl red and the Vogel-Proskauer test. So there is a broth, which is the MRVP broth, and you can use it to test for two different fermentation products. So usually in the lab, you either take 2 tubes of the same broth, or you divide the broth in 2, and you make these 2 different tests. So the methyl red, the MR test, tests for acidic fermentation products. And what you do is you culture your bacteria in the MRVP broth, and then you add methyl red. And methyl red is a pH indicator. It's not exactly the same as phenol red. It's also a pH indicator, but the range is a little bit more in the acidic part. So it changes color when the medium is very acidic. So if you add methyl red and the broth is red, that it means a positive reaction, as you can see in the figure to the right. The VP test is a test for neutral fermentation products. So if you get a bacterium that is MR positive, usually is not going to be VP positive. They go opposite because the bacterium is either doing more acidic products or more neutral products. But it's always a good thing to test for both. So the VP reagents are 2. VP1, VP2, it's alpha-naphtol and potassium hydroxide, and when you add it, you are going to observe this dark reddish brownish color if it's positive. There was also a mention in the section of amino acid or protein catabolism of amino acid degradation test. So there are a variety of those. And what they test is if the bacterium has the ability to break down certain amino acids. And when I say variety, is because they are going to test for different reactions and different amino acids. So for example, decarboxylation means that the bacterium is able to remove the carboxyl group that's the acidic group, from an amino acid. And one of the most common one is called the ornithine decarboxylation. The other one is the deamination reaction. So in this case, the group that is removed is the NH2, the amino group. And a common one is called the phenylalanine deamination reaction. And these media tend to have that specific name. So Mannitol-Salt Agar is going to test for phenylalanine deamination. Then we have the hydrogen sulfide production test. So there are a couple of amino acids that have sulfur in them. And when that sulfur is removed, then you get the production of hydrogen sulfide, which is a gas, is H2S. Now, hydrogen sulfide is a very stinky gas, but if you just let it go, it will go. So the media, what they have is chemicals that react with that hydrogen sulfide, producing a black precipitate. So you can see it to the left. This is a tube that gets black. So the black means it's a positive reaction for hydrogen sulfide. It doesn't mean that it is hydrogen sulfide. It tends to be an iron salt of sulfide when the medium reacts with the H2S gas. A very important reaction is the Indole reaction. So when you were looking at the Unit 3 slide, there is a reference to a panel of tests called the INVIC test. And these are very useful reactions, for example, when you need to discriminate between what we call enterics. Enterics are a group of gram-negative bacteria. E. coli is there, Salmonella is there, a few others are there. They are all gram-negative rods, and they are very commonly in the GI tract. That's why they're called enterics, and they can cause different gastrointestinal infections. So with that test, if you look at the table in the original slides, you can see that it's very easy to differentiate between those bacteria if you do the whole panel, because it's hard to find two that has the same reaction for everything. So indole is one of these, and this is also about amino acid metabolism. So there is amino acid called tryptophan, and if the bacterium is able to degrade tryptophan, it's going to produce indole. And there is a reagent called the Coagulase reagent that if you add it to the medium in the presence of indole, it's going to turn this very bright red. So the indole reaction is red, positive when you add Coagulase reagent. In the part that is dedicated to tests related to respiration, we talked about catalase and we talked about oxidase, and I want to also talk about nitrate reduction. So it may have been mentioned especially in the lecture part when you studied cellular respiration. Cellular respiration is a very broad catabolic, so degradation pathway, and at the end, the electrons that are being removed as the original product, can be glucose or something else, is being oxidized, and electrons are taken up by oxygen. That is the most common, or the ones that most students know, the cellular respiration. At the end, oxygen is the final electron acceptor. However, some bacteria are able to do what is called anaerobic respiration, not to be confused with fermentation. So this also involves the electron transport chain, but instead of oxygen, there are other substances that can take up those electrons. And some of these can be nitrates. So nitrate reduction is a pathway. It could go through different intermediates. So nitrate, which is NO3, can be reduced to nitrite, which is NO2, which can be reduced to nitric oxide, to nitrous oxide, and all the way to nitrogen gas. So many bacteria have the ability to reduce nitrate, but they do it differently. So some of them may only go nitrate to nitrite, others can go all the way to nitrogen gas. So how do you know that? There is this test. The nitrate reduction test has two reagents, nitrate A and nitrate B. So you add A and B to your test, and then if it turns positive, then that means that the nitrate has been downgraded to nitrite. However, sometimes that nitrite has been reduced all the way to, let's say, some of those oxides or even nitrogen. So if you add A and B and it's yellow, so it doesn't change color, then you want to check if it has been reduced further. And then you add the third reagent, which is a zinc powder, in which case it's the opposite. If it's red, then it is a negative reaction. So let me just go over really quickly. You add A and B, and if it's red, it's positive. If it's not red, then you still don't know, and you add a third reagent called zinc. And when you add zinc, red is negative and yellow is positive. So again, this is a way to measure the ability of a bacterium to degrade nitrate. And there are also a number of tests that look at the ability of bacteria to degrade proteins. So I think what is mentioned there is gelatinase, and gelatinase means that the gelatin can be broken down, and that means that you add it to a gelatin cube and then it becomes liquefied. Well, coagulase tests for the capacity or the ability, it tests for the presence of an enzyme called coagulase. So coagulase clotting. This is an enzyme that clots fibrin in the blood. And when you think about it, if you have a bacterium that is in the blood, that means that that bacterium could be attacked by the immune system. But if they clot the fibrin, a little bit like replicating what would happen during blood clotting, that produces like a protective cocoon around the bacterium, so that protects us. So coagulase positivity means that that bacterium is more harmful because it can hide from the immune system. And this is very typical of Staphylococcus aureus. How you test it? There is a reagent called coagulase plasma. It's exactly what the name says. It's a plasma, like the liquid part of your blood. If you add the bacterium and it clots, so it becomes kind of gel-like, and you can see it in the picture. You tilt the tube, but the reagent is jelly, it's not flowing, then that it means that it's a positive reaction. So again, you want to review these additional slides because they add to the topic of Unit 3. So in Unit 4, we are going to look at quantitation and isolation of bacteria. And I think I mentioned it last week that some of the material that was needed for the Labster lab, we are going to see this here in a little bit more detail. So how do you quantitate bacteria? There are a number of ways to do that, but the one that will be seen in the simulation, or one of them that you will see, is what we call plate counts. And it's exactly what the name says. So you put your bacterial suspension on a petri dish, on an agar plate, and then you count the colonies. And why is this? What's the advantages of this method? Because you are counting only living bacteria. Because you may have, a very thick suspension of bacteria, but half of them may be dead. So the only ones that are going to produce colonies are the ones alive. And it is a powerful way to do that, but it also takes time because you need at least a day or two in order to count the colonies. Now I'm going to just go to this next one and then I go back to explain more in detail. So let's say that you have a completely unknown bacterial suspension. Especially think about if you're taking a sample that by definition you're going to have a lot of bacteria. Let's say a fecal sample, for example. If you just take that and swab it on a plate, you're just going to get too many colonies. It's going to be impossible to count because they are going to just grow on top of each other, producing what we call a lawn of bacteria. So how do you know how to dilute that suspension that you want to quantify if you have no idea how many they are? So that's why we do serial dilutions. So it's a way to dilute the sample. So at some point you're going to get a countable number of colonies. And two things. One is, so what dilutions you do? Well, once you know your sample very well, you may know what kind of dilutions work. But if you have a completely unknown sample, you may need to just have a broad range of dilutions so you are sure that one of them is going to work. So, for example, in this particular diagram, you can see that you start with 1 in 10 and you go all the way to 1 in 100,000. And you will see, as you can see on the plates at the end, that you will go from a lot to very few or none. And you will be looking for a number of colonies that can be counted, but they are enough not to be just error in the system. So usually a good number is around between, let's say, 50 and 150, or up to 200 sometimes. So it's a countable number. You don't want very few, because very few could be just a contamination or a manipulation error. So that's why you dilute, and that's why you do this wide range of dilutions. Now, why serial? Well, look at those two numbers at the end, at the either ends, right? 1 in 10 is very simple. You take one part and mix it with 9 parts of solvent. It could be media, it could be water, etc. And that will give you a 1 in 10 dilution. But if you're making a 1 in 100,000 dilution, that would make 1 part of your suspension and 99,999 of the solvent. Well, that is impossible to do in manageable volumes. So the easiest way is to do in a series. So you start with 1 in 10, and then you move from 1 in 10 to the next, and to the next, and to the next. So you're making 1 in 10 steps along the way, so that way you can reach very high dilution numbers, but you are not manipulating weird, very small or very large number of volumes. And so again, you will count the colonies, and in this example, 71 is a good number, because if it's in the hundreds, it's pretty much impossible to count, and also it's not reliable, because colonies may be on top of each other. And if it's a very low number, like 6, that's too few and could be, it's not reliable. How do you calculate the original number? Well, you will multiply the colony count by the dilution factor. So let's say 71 was in your 1 in 1,000 dilution, so you multiply 71 by 1,000, so that would be 71,000 bacteria per mil. Please note that you are adding to the plate 1 milliliter, which will give you at the end bacteria per milliliter. Let's say that you added 100 microliter, so that would be 0.1 milliliters to the plate, then you are doing an additional 1 in 10 dilution. So depending on the volume you add, you may need to factor some more dilution factors along the way. Okay, so again, if you have 71 colonies on the plate, but you only inoculated 0.1 mils from your 1 in 1,000 dilution, then you have to multiply 71 by 10. That's the dilution factor between 0.1 and 1 milliliter, and you have to multiply it by 1,000, which came from the original dilution. So in that case, you would have 710,000 bacteria per mil. Okay, let's go over how do you obtain pure culture. So microbiologists can start from a very complex sample that has multiple bacteria, and eventually they are able to separate those bacteria into pure cultures. And the best way to do that is the easiest way to do using what we call a streak plate. And in the simulations, you do streak plates, and what it entails is that you dilute your sample on the plate. You keep spreading your suspension on the plate, so by the 3rd or 4th, sometimes the 5th streak on the plate, the sample has been diluted enough, so the colonies are going to be distinct. So for example, in this diagram, you see E. coli and Micrococcus luteus, and the colonies are different. E. coli are a little bit fuzzier. M. luteus tends to be a little bit yellower and smaller, so you can then pick one colony of each and then streak or re-streak on a different plate, and that way you repeat the process a few times just to be sure that you don't have any contamination. Very often in microbiology, you are going to see the report of, so reporting the number of bacteria as CFUs, colony forming units, instead of colonies. And this is just a different name that reflects that when you are counting a colony, you assume that those are cells that are coming from a single cell. However, there may be 2 cells or 3 cells that are so close to each other that when they divide, you can only observe one colony. So colony forming unit is a little bit like to be safe, that you are not saying this is coming from a single cell. It could be a little bit more than that, but we cannot really tell. Okay, so here is a diagram, and I do recommend that you go and watch the videos. How is this done in real life? You can start from broth or a solid medium. You flame or heat up your loop. You wait until it cools down, and then you take a little bit, either from the solid or the broth culture, and you streak on a quadrant. And when I say quadrant, some people do quadrants, other people do what it's called a T on the plate, and then you get 3 zones. So you can call it quadrants or zones, but the main thing is that you're going to have different zones on the plate. Okay, so we are in step 3. You streak the loop in a zigzag pattern on one quadrant of the plate. Now comes the important part. You have to flame your loop. And the reason is that you want to spread the cells that you already put in that first quadrant without adding more. So by flaming the loop, you are killing everybody else on the loop. You wait until it cools down, and then you drag the loop from the first quadrant into the second one, and then you keep zigzagging. So this is why we say you are diluting the cells on the plate, because you took a little bit of sample from that first quadrant and spread it to the second. You repeat the procedure at least two times more. You flame between the quadrants. Again, the importance for that is that you want to dilute your cells. So you want to kill the cells on the loop, and you keep moving a little bit from the one quadrant to the next and then to the next, and that way you are just spreading less and less cells on the plate. And if you did it correctly, then you will see this pattern when then usually the last quadrant will show completely isolated colonies. So with this now, let's move on to special media, because when we're talking about streaking, and you may have, a very complex sample, and yes, you can thin them out and separate them using streak plates, but sometimes you know what you want, and you are looking for a specific group of bacteria, so you can a little bit, fasten the whole process. So speed up the process. So we can have, for example, we can use differential media. If you kind of know that your sample may have E. coli or a certain characteristics of a microbe, differential media is going to make them look different on the plate. We are going to see examples of that. Enrichment media, I don't know why this is repeated. So enrichment media enhance the growth of the desired microbes with added chemicals. Let's say that you are looking for a bacterium that is able to degrade, let's say, natural gas is not a good one. Let's say some kind of poison. Okay, so you are testing for bioremediation, and you want to look for bacteria that are able to degrade some kind of environmental poison. Well, how can you find those bacteria? You add the whatever that substance is to your medium, which is going to help the desired microbes to grow. Then you have selective media. So let's say that you are looking, and this is very commonly used when you are looking at enterics. You're talking enterics, those are the ones that are common in the GI system, and they tend to be gram-negatives. So if you are trying to figure out the diarrhea of a person, if it's due to E. coli or something else, you can say, okay, I don't care about gram-positives. I just want to isolate the gram-negatives. So you may have a selective medium that will depress the growth of gram-positives, so that only the gram-negatives will grow. And we will see a few examples of media that can be both selective and differential. Okay, let's start with one. Mannitol-Salt Agar. This is both selective and differential. It is selective because it contains salt, right? So only salt-tolerant bacteria will grow on them, and you may recall that that has a name, halophiles, H-A-L-O. Halophiles are bacteria that either like it salty or are able to grow on salty, in salty environments. And there are some bacteria that are obligate halophiles, the ones that, I don't know, Salt Lake or Dead Sea, they do require the high salt content, but others are what we call facultative halophiles. So they may not like it very much, but they're able to survive. Staphylococci, so gram-positives, and especially some staphylococci, are more resistant to high salt content. So Mannitol-Salt Agar, MSA, is particularly good for gram-positive bacteria. Now, the differential part comes from mannitol. So mannitol is a sugar, and if a bacterium is able to ferment mannitol, then that means that they are fermenting, so the pH will become more acidic. And there is phenol red in the medium. It's the same phenol red that you have in phenol red broth. So the mannitol fermenters are going to give you yellow colonies. So, for example, when you have Staphylococcus aureus, if you want to differentiate Staph aureus from other staph, MSA is useful because they are able to grow and also to ferment mannitol. Next example is blood agar, and this is a differential medium. And the name blood is because there is blood in the sample, in the medium. It contains sheep blood, so there are red blood cells inside the medium. Certain bacteria are able to break down red blood cells, and this is what we call hemolysis. And spoiler alert, usually the more hemolytic bacteria are, the more dangerous they may be from a disease point of view. So there are three kinds of hemolysis, alpha, beta, and gamma. And gamma hemolysis is basically no hemolysis. So the bacterium is unable to break down the red blood cells. As you can see to the left, you see the colonies, but there's nothing special about them. But if you compare beta and alpha hemolysis, alpha hemolysis is a partial, incomplete degradation of the red blood cells, and you're going to see this greenish halo around the colonies. And beta hemolysis is a complete degradation of the red blood cells, so you will see this clearing, these clear halos around the colonies. And when you're doing, for example, throat culture, streptococci are very common in the throat. Two very common streptococci are Streptococcus pneumonia and Streptococcus pyogenes. Strep pneumonia, well, the name indicates it can cause pneumonia. S. pyogenes causes strep throat. They also can cause all kinds of sepsis and complications. Strep pneumonia is alpha hemolytic, strep pyogenes is beta hemolytic. So by culturing it on blood agar, you can differentiate between the two. Then we have MacConkey Agar, and this is another selective and differential medium. You saw that in the previous simulation. So it is selective because it has the ability to inhibit the growth of gram-positive bacteria. So only gram-negative bacteria can grow on MacConkey, and this is because of the content of biosalts and crystal violet. And then it's differential because it also contains lactose. So again, we are looking at the fermentation product here. Those that are able to ferment lactose will change color, and the colonies will be red. And if they are not lactose fermenters, then they are going to be these pale yellow, very pale yellow colonies. The pH indicator here is neutral red. So as you can see, we have a bunch of different reds, phenol red, methyl red, neutral red. So just be clear which test uses which, but they are all looking for change in pH. Then there is this next one. It's called the Eosin-Methylene blue agar, or EMB agar. This is also selective differential. It's kind of similar in what it does to MacConkey. So it's also selective for gram-negative rods because of the presence of methylene blue. And it's differential for lactose fermentation. And what is kind of different is that the colors. So in the case of EMB agar, you're going to see beautiful colors. And the picture really doesn't do justice, but for example, E. coli is going to produce this green metallic color colonies, very typical. Others that are less fermenters may produce kind of a brownish, pinkish, reddish color. But the ones that are non-fermenters, they won't provide any special color. And with this, this would be the end of the Unit 4 lecture. Thank you.